Oi, tudo bem? Um prazer estar aqui para apresentar para vocês. Antes de começar, a primeira coisa que eu gosto sempre de falar é que eu respondo, vocês podem perguntar em português, mas a minha palestra vai ser em inglês. Eu já estou nos Estados Unidos há mais tempo eu morei nos Estados Unidos mais tempo que eu morei no Brasil. E como eu digo, eu não sei ciência em português. Então é melhor para vocês se eu der a palestra em inglês. E também como eu moro nos Estados Unidos há muito tempo, mas o nosso mundo de ciência é muito pequeno. Então eu quero dar uma introdução da onde que eu saí do Brasil. Então eu moro nos Estados Unidos desde 1989. Eu vim fazer a minha faculdade aqui desde o começo, em Washington, mas a minha família mora em Brasília, agora ainda mora, meus pais, meus irmãos, e o resto da minha família, tios, avós, moram em São Paulo, na área do Vale do Paraíba, de São José dos Campos. Eu também morei um bom tempo no Sul, em Santa Maria. Mas então, é sempre bom ter uma conexão com o pessoal do Brasil. E a palestra que eu vou dar agora vai ser o tópico de Magnetic 3D Bioprinting, ou, como eu digo, a cultura celular a cultura magnética em 3D. Então, agora eu vou trocar de português para inglês, mas vocês podem perguntar as perguntas em português e eu respondo. Tá bom. So, now I'm going to talk about magnetic 3D cell culture, which is, I'm going to give you a little bit of where the technology came from. Then I'm going to go into the nuts and bolts, details of how the technology works. And at the end, I'm going to show you some results, some published, some unpublished. And at the end, I'm going to show some new things we're developing that it's a new way of look at biology. And at the very end, I'm going to plant some seeds on how you can use magnetic 3D cell culture for maybe with COVID-19, which is as we know, it has transformed our lives in the last few months, and we're trying to find new ways of apply our technology also for viral production, antiviral testing, and vaccine production and other related topics. All right, so where the technology came from, the whole work that I'm going to present started in a collaboration between myself when I was at the Anderson Cancer Center and a few friends at Rice University. We started this work about 11, 12 years ago. Then we patented the technology. We spun off a company, which we call Nano 3D Biosciences. And as of 2 years ago, Greiner Bio-One acquired the technology. And that's why now I'm with Greiner. And I come to Brazil to work with my Greiner colleagues. They're excellent. The factory of Greiner in Brazil is in Americana. So, of course, if I'm going to talk about 3D cell culture, especially that this is it's a broad conference, I have to give you a little bit of a high-level view of why 3D cell culture is important. We know how to culture cells. We've cultured cells for 100 years. We take cells, we put in a petri dish, they stick to the bottom, to the plastic, and they grow. But today we understand that cells, when they grow on a plastic surface, which you call 2D cell culture or monolayer cell culture, they don't represent how cells grow in vivo, in the body. And this has a lot to do because cells in the body, they interact with each other in 3D. They have different cell types that also interact within the 3D environment. And we know that this 3D aspect, the cell-cell contact, the 3D space is very important to replicate in vivo biology. And from a more economical point, we all know that developing drugs is very expensive and takes a long time. And this is a good example why 3D cell culture is very relevant. And I'll show you some real results to in a way confirm our hypothesis that 3D cell culture can improve drug development. So drugs are very expensive to develop. They take a long time, and many reasons that it's expensive, it takes a long time, is because cells, we start usually the drug screening, the drug development work, we started with thousands of compounds and screen the compounds on a monolayer culture, again flat, which is not a good representation of in vivo biology. Then we pick some compounds and we pass the winner compounds to animal models, which are not, doesn't represent human physiology. And when they get to humans, most drugs fail from poor efficacy or toxicity. So we are trying to use 3D cell culture to improve the in vitro biology with in vivo results, to make it faster, cheaper, better. And as we all know, drugs don't take a long time. In today's world, when we have the COVID-19 and we want a drug developed yesterday, it doesn't work like that. And it's not because of regulatory process. We all know that. It's because the process is challenging. We rely on, proving hypothesis to learn and develop the right safe, efficacious drugs for us to use. Okay, so, but now you go from a practical point of view. So we have this paradigm shift where we switch from monolayer culture to 3D culture. And one thing that we realize when we shift from 2D from 3D is how important cells attached to plastic is for all the workflow, for all the manipulation. Cell culture is easy because cells are attached to plastic. And that's a realization that comes very quick when you start working with 3D cell culture. So what we do, we wanted we develop a tool that is a surrogate for cells attached to plastic. So, and as a good example, changing media, doing immunofluorescence staining. Why is it easy? It's easy because on monolayer culture. Why it's easy? Because cells are stuck to plastic. You can remove the media, add the media. You can, when you're doing immunofluorescence, you can block your cells, wash your cells, and that's very fast, very easy. But when you go to 3D, you lose that capability. You cannot manipulate the cells very, very easily. So we use magnetic fields as a surrogate for cell attachment to make 3D cell culture in a similar workflow as we have with monolayer culture. So in this case, of course, cells are not magnetic, so we need to magnetize the cells. So this is, we have a nanoparticle assembly that is iron oxide, gold nanoparticles, and polylysine. We call it nanoshuttle. So it would just add to the cells, electrostatically adheres to the cell, and you can use cells in suspension. Cells, we have protocols to take cells from the body, break it apart, and magnetize them, and I'll show some of the results. And once you magnetize the cells, which is a simple step, you can detach whatever method you use, and you can put a magnetic field on top, which we call the levitation, which actually levitate the cells and they grow in 3D. This is it's a lower throughput. It's 6 and 24, and I'm not going to get into much of the levitation work today. And we have the bioprinting. The bioprinting, we use a cell repellent surface, and we have a magnetic field on the bottom. And we can use the magnetic field to manipulate the cells, to pattern the cells, to layer cells, and we can go in very high throughput. This we can use from 6 well all the way to 1,536. And this is just a quick tour from some quick results. And because we can bring cells together fast, here you have fibroblasts. We just bring them together and they make spheroids very rapidly. This is our fibroblast. And this is on the right, top right here is a co-culture of glioblastoma GFP labeled with normal human astrocyte MTR label. We actually culture them separately, and then we magnetically guide them together. And it's cool that you can actually see the glioblastoma GFP label to crawl and completely invade over time the astrocytes. And if you're studying metastasis, which we always wanted to study metastasis if we're dealing with cancer, researching cancer, this is a really cool tool. On the bottom right is a co-culture of pre-adipocyte stem cells and endothelial cells. The red is pyrolypin, which is a marker for adipocytes, and the green are GFP endothelial cells. And you can see the endothelial cells are sprouting within the culture. This was actually the cover of Tissue Engineering issue when we published. And there are a couple of things that I like to mention here. So when you go to 3D cell culture, we often mention adding complexity to the tissue, culturing different cell types, but there's no magic. You have to optimize. And this work was important because when we start doing this, trying to get this adipocyte structure with the endothelial cells sprouting within the structure, it wasn't easy. We had to optimize the ratio of cells. And for us to see the endothelial phenotype here, we have to culture we have to use 95% pre-adipocyte stem cells and 5% endothelial cells. Otherwise, you didn't see this problem. And the staining of this culture, this is a confocal image. And the reason this image came out so nice is because we stained this whole mount, which means we put a magnetic field on the bottom, and we're able to wash and without having to paraffin embed or freeze this section. So since I'm in industry, I always like to mention that still, when you're in the scientific world, the best marketing tool or the best marketing is a good publication. So when we left in the industry, starting in the 3D biosciences, we worked really hard to publish in good journals. And we're very successful doing that. But another reason that I mentioned the publication list here, and these are not everything, it's just some of them, is because we use nanoparticles. So you're probably asking, what do the nanoparticles do to the cells? And these journals always ask. And we have published and we have shown that the nanoparticles that we use are bioinert. They don't do anything to the cells. And we have a 5-page white paper. We've compiled all the results on the website that we compile our results. And we look at viability, proliferation with or without nanoparticles, protein expression, gene, stem cell differentiation, inflammatory response, inflammatory stress, oxidative stress, gene expression, and there's no difference between nanoparticles and non-nanoparticles. And we are also going to show some results. This is work we've done in collaboration with the Scripps Research Institute, where we did a high-throughput screening. Again, I'm going to show the results. But before we start working, they started working with us. They also validated that the nanoparticles don't do anything. So we picked a pen of cell lines, a pen of the control compounds, and we compared nanoshuttle, non-nanoshuttle with the control compounds, looking at dose dependence. And as you can see, this is our dose dependence curve that we measure IC50. On the right with nanoshuttle, the left no nanoshuttle, and there's no difference between the curves. So the nanoparticles are also bioinert as far as drug interaction with the cells. So the technique is very simple. And here's an example how we make a spheroid. We have a pin magnet right in the middle of a cell repellent plate. You put the plate on the magnet, and as soon as you put the cells on the well, they come together. You do not need to leave the cells on the magnet. Most applications, just as few minutes as 15 minutes, it's enough. You remove from the magnet, and cell-cell interaction makes the spheroids. So here's a quick movie that'll show you the exactly workflow of the technique, which is good for you to see. So it's a kit, the bioprinting kit. So first thing, you add your nanoshuttle to your cells, and the cells will stick overnight, generally overnight. You wash your cells, the free nanoparticles go away, you detach, you count, and then you're going to take your cell repellent plate, put it on the magnet. So you count everything, same workflow as you use. Take your cells, you dispense your cells. See the magnet, right in the middle of the well, you dispense your cells. This is a flat bottom plate too. So magnetic field brings the cells together. So within 15 minutes on the magnet, see the cells are right in the middle. And then overnight, cell-cell interaction takes over, you make a spheroid. And again, this is a flat bottom. Now you want to change media, you want to add a compound if you're doing drug screening. You put on the bigger magnet so it holds the spheroids tighter. You take your media out, you add fresh media, and that's it. So now you add your compound, and it's the same workflow. So the magnetic field really is a surrogate for the cell attachment. And here we show we have 96,384 all the way to 1536. And we call 3D and a 2D workflow. So, and now this is, of course, if you're dealing with spheroids, you got to show reproducibility. This is our 384 system. In 600 cells, 20,000 cells, flat surface, and you can see very reproducibly sized. And this is application that we published at Prometheus PubHub. And now you have your number of cells, diameter, and you can see very tight error bars, so it means very reproducible spheroids in size. And then you have your number of cells and viability using cell titer glow, very linear for a wide range of cell numbers, which is what we need. And again, this data you can find on Prometheus PubHub and on our website too. So next is it's one way of, I think, of magnetic 3D cell culture. Magnetic 3D cell culture is an enabling tool. What I mean by that, you magnetize your cells and you can do different things. In this case, we want to create more than one spheroid per well. So we took a 6-well plate and we took a 384 magnet drive, and I'll show you a picture of what I mean. And we make several spheroids of the same size. So here you have 20,000 cells per spheroid all the way to 600. And basically what we did, we took a 6-well plate, cell repellent, and we put it on a drive we call magnet. It's a magnet array that you have about 50 magnets sitting under each well. And now you can make very reproducible spheroids. So these are spheroids from the same well, each column. So it's an enabling tool to facilitate and manipulate. And in many applications, you may want to generate many spheroids per well. So, of course, I love this movie because it's always nice to see movement. So this is iPSC cardiomyocytes, induced pluripotent stem cells, and we printed in a 384 well flat, and watch what the cells do. Sometimes Zoom doesn't, there's a delay, but they beat very nice periodically if you don't see on your movie. But in this culture, beat it regularly on the same pace for 20 days. So, and the beauty of the magnetic field, you can actually hold the spheroids and sorry if I move it. I'm not sure if you're seeing my face on the recording, but so I'm going to move out of the way just so it's more important the data than seeing me. But we also got this work was done with AstraZeneca from England, did this work. They actually magnetized the cardiomyocyte spheroids and were able to guide the spheroids onto electrode to generate to make electrophysiology measurements. If you have worked with cardiomyocytes or cells that you need to do electrophysiology, it's not trivial. It's just about impossible to use 3D cell culture with microphysiology. And here, I went to the wrong way. And here they placed the spheroid magnetically on the electrode. Basically, you just have the electrode, and you guided you have the electrode, and you guided the spheroid magnetically. They stick to the electrode, and you measure. Here they measure the impedance, field potential with magnet, very regular beating. And without magnet, it just doesn't work. So this was very nice work done. And was shared by AstraZeneca. So now I'm going to switch gears and go to cancer application, which is most of our work, but I'll show you some different cell types later in the presentation. So cancer cells, and I like to show this slide to continue the topic of magnetic 3D cell culture as an enabling tool. So by magnetizing the cell, you can facilitate doing things that are not trivial on a flat surface. Why do I keep mentioning flat surface? Because flat surface, if you want to do imaging, high resolution imaging, flat surface is very important. So it's relevant. So here, what we did here we are, it's a collaboration with MD Anderson, and we're looking at CAR T cells targeting. And we use NALM-6 cells. NALM-6 are leukemia cells, which means they don't generate 3D structures. They are blood cells. So why do you need a 3D cell culture here? So what we did here, we brought the cells together magnetically in the middle of the well to detect easily how, if the CAR T are specifically targeting the cells. So what we did is we took the NALM-6, the leukemia cells, we had green cells, which are CD19 negative, we had red cells, which are CD19 positive, and magnetized them. Then we brought, with the magnet, we brought them all in the middle of the well. And you can see here on the right, they're all red and green. Then we added a CAR T cells not magnetized. We just added to the well. And this CAR T is supposed to target the red cells, the CD19 positive. And you can see overnight, the CD19 goes away. Actually, no, sorry, 48 hours, the CD19 goes away. And the ratio of CAR T to target cells was very low, which is very good for us to detect this, the killing. And again, it didn't kill everything. If you zoom in on these cultures, you can see there's some leftover cells there too. So again, it's specific. We're able to detect the CAR T application without having to scan and using chromoassay. Chromoassay is nasty and it's not specific. So this is, now I'm moving towards the bio 3D cell culture. And in this work we did with Jeff Bartholomeus at MD Anderson. And we actually compared the PDX, the in vivo, with the magnetized culture. This was a triple negative inflammatory breast cancer. So we took the tumor from the patient and put it in mouse, and we grew the tumor. Then we took the tumor, we split it in half. In half, we processed the standard procedure. The other half, we broke the tissue apart, magnetized the cells, and brought it back together. And here are the results. So we look at icadirin, Vimentin, Ki67, and Pismad. And actually, this is a better slide. So here, what you see here is, sorry, my mouse is not working now. Hold on, I got it. So the left here is the in vivo, the tumor. It's a paraffin embedded tissue and the immunohistochemistry there with the different markers. The exact same thing we did with the bioprinted samples. So the middle and the right these are in vitro tissue, bioprinted tissue, and the left is the in vivo. And you can see the marker distribution between the different markers is pretty much the same. So, and on this work, we also did the gene expression comparison between the vivo and the 3D culture, and it was a 95% correlation. We did dose response, and it worked really well. So, and then of course, when you do a PDX, this is a prostate cancer PDX we did as a collaboration with UT Medical School, University of Texas Medical School, where with a ½ gram of PDX sample, we could break in hundreds of cultures. And within 96 hours, you could test a panel of compounds. Here I showed just the dose dependence curve for 4 compounds. As you can see, very nice curves for the different compounds. And of course, we can put it back on the magnet and do the immunohistochemistry for the tumors. And part of the goal of magnetic 3D cell culture also is looking into personalized medicine. People are using organoids, spheroids, and here was a it's a culture from bone biopsy from a prostate cancer metastatic site. They did a needle aspiration. We broke the tissue apart, removed the bone, broke the tissue apart. There was some bone in the culture. We removed, magnetized the cells, brought them back together, and within 96 hours, we're able to get a very nice IC dose dependence curve for doxorubicin, which is the drug that it's used for the treatment of this cancer. And of course, this cancer was EpiCam positive. We're able to detect by just putting back in the magnet, staining, which is the marker for this type of prostate cancer. So these are some of the personalized medicine applications. And I'll continue in that topic, but also bridging into high throughput screening. This is a work that was funded by NCI, NIH, the National Stand Cancer Institute from NIH. And it was a collaboration between Scripps, myself, or Nano3D Bioscience at the time, in Cold Spring Harbor. And this work was to validate a high throughput screening platform. And we used pancreatic cancer. So this is just to show you the system, how the magnet drive for the 1536 look like. As you can see, it's a very dense array of magnets. These wells are very small. And just to show the power of the magnetic field, if you put the cells in the well, as you can see here, without the magnetic field, they all dispersed in the well. Now you place the magnetic field, they all come together. And this is a colon cancer cell line and lung cancer cell line. So this is the beauty of the power of the technique. Bring these cells together in a reproducible way. And here it's a picture of a 1536 well that was assembled. And you can see on the 1536 plate, using 1,200 cells per well, you can see very reproducible size, and one thing that is important too, which is I'm going to get into next, is the positioning of the spheroids within the well. So as I mentioned before, when you're doing 3D cell culture, looking at a high resolution image is very important. And or it's very desirable, especially when you're going to high content screening. And for you to do that, you cannot use the round surfaces, which a lot of the work being done is using round bottom. But when you want to do a high resolution image, that just doesn't work. You need a flat surface. So, but when you're using a flat surface with spheroids, as you can see here on the left, the spheroids over time, because of thermal motion, they tend to move. And once the spheroid gets to the side, it's very hard to image the spheroids. And so if you look at this plate so every dot here is a spheroid. So if you look at this plate on the left, you lost about a quarter of your data because the spheroids move. But with our system, with the magnetic 3D cell culture, you can put them back on the magnet and you can recenter the spheroids. Now, not only you didn't lose your data point, but also you position the spheroids in a place that your microscope can go and visualize very easily. And remember, when you're talking about high throughput screening, you may think this is not, it's not needed. Of course it's needed, because you don't want to lose a quarter of your 384 or 1536 plates. Those are a lot of samples. Next, if you're doing high throughput imaging or high content imaging, your microscope is going to scan. So if you have to look for your spheroid in the well, it takes a very long time to image your plate. Now, if your microscope knows where the spheroid is, it can move very fast. So it is meaningful, especially when you're screening thousands of compounds. Sorry. So, and of course, we're talking about 3D cell culture. It's very important to show you the morphology. And this is one point that I always tell people, because some people start doing 3D cell culture and they're like, oh, my cells don't form a nice spheroid. No, not all cells make nice spheroids. That's just the biology. And I tell you, sometimes they look like they make spheroids, but they don't really. They're taking the shape of the well if you're using a round bottom plate. But here, again, we are using here, the first cell here is ovarian cancer cell line. I'm going to move it here. This is ovarian cancer cell line, pink one. The first column here, you're just printing the cells, and you can see all the cells right after you print, right in the middle of the well. And if you look at the cell lines, HT29 in pink one, HT20 ovarian cancer cell line, pink one is pancreatic cancer cell line. They make beautiful 3D structures, okay, after 72 hours. Sorry, I'm speaking fast. So now it's important for me to break down the rest of this data, because this goes into the personalized medicine aspect of our tool. In here we have 4 cell types here, AGM1 and AG21. Sorry, my wife is going to give me some water, but I locked the door. That's okay. So AGM1, H21, AGM1 calf, H21 calf. In AGM1 is one patient, H21 is another patient. AGM1 calf is the cancer-associated fibroblast from the same patient. And again, so we have pairs here, AGM1 and AGM1 calf, basically it's the same tumor. We separated the cancer from the fibroblast, and we cultured them separately. Same thing for AGM1, okay? But what is interesting, if you look at the cancer-associated fibroblast, you bring them together, they make a nice spheroid. You go to the cancer cells, they don't. They make organoids, which there is a difference between organoids. Usually they're generated from stem cells. They're differentiated for very specific structures where the cells are polarized. And there's no matrigel here, because usually use matrigel to create the organoids, but don't. And we characterize them. We don't need to use matrigel. And even by bringing the cells together, it didn't matter. They made organoids. So great. So, but still, we magnetize them. We could still use all the benefits of magnetic 3D cell culture. Okay. So the results of this work. So one of the hypotheses we had for this when we started this work is first to compare 2D versus 3D, and to demonstrate or to verify the hypothesis that 3D has less false positives than 2D on a drug screen. And that's, remember, very early on my motivation slide, I said that most drugs, when they get to humans, they fail from a toxicity or poor efficacy. So this is what we're going to try to do to show here. So how we went about to show that. We used the FDA library. So 3,300 compounds, FDA library, which does not have cancer prescribed drugs. If there isn't cancer prescribed drugs, one would assume if it's killing the cancer cells, it's because it's a false positive. So it's the best we could do, let's put it this way. So when you look at the results, break it down, the results, 3D always showed less hits, less compounds that killed the cancer cells than 2D. So we, in a way, we proved our hypothesis. Yes, you can reduce the number of false positives when you're using 3D. So another way of looking at these results is actually looking at this plot here, which is inhibition in 2D versus inhibition in 3D. Every dot here is a compound, okay? So if the dot is on the white quadrant, means that it didn't do anything to the cells expected. The pink are cells, drugs that are only active in 2D. The green is the overlap, 2D versus 3D. There were very few compounds that were even active in the 3D, but there was one compound that was significantly active in 3D, which did not kill the cells in 2D. And this is disulfiram. And which was cool is that disulfiram is actually phase 1 clinical trial. So we picked the needle in the haystack. So that's, we not only showed that that 3D reduced the false positives, but it can actually pick the good compounds as well. And we've done 150,000 compound screening, 2D versus 3D used in 1536. This will be published, but if you do compound screening, I like to bring up, point to you that the Z prime of these essays, which is the measure of the standard deviation between positive and negative, number of standard deviations, difference between the positive and negative controls, is 0.72. And if it's above 0.5, is an excellent assay. And this was done cell titer glow, so it's using chemiluminescence. So now, as I mentioned, we took the patient tumor, and we separate the caps from the cancer cells, right? So, and we test them separately. Now we want to bring them back together, co-culture them. So what we did, we labeled the caps red, the cancer cells green, and we mixed the two of them. So as you can see, the caps, the cancer-associated fibroblasts, made beautiful spheroid. The cancer cells made the organoids as expected. Now, the co-culture, what was interesting, look at 24 hours. The cells all came back together, and the cancer cells come out of the culture. So it's important. Biology, cancer is a dynamic process. So time point is very important. So the cells interact together, the cancer came out, and that's, again, metastasis is mobility, is cell migration. And we also test the drug sensitivity between these three systems at 96 hours. The caps actually were much more sensitive to the compounds than the co-culture and the monoculture. And the monoculture and co-culture showed very similar results. So from this point of cell mobility, I'm going to go towards the next part of my talk, which is a new way of looking at biology, which is, can we use cell mobility to understand, as a new tool to understand biology? So why do I bring that up? Because when you magnetize the cells and bring them fast on a flat surface, the cultures move, the cells move a lot. And in our hypothesis, can we use the cell movement, the cell-cell interaction, if it's collective or not, as a readout of biology to, fingerprint cell types based on this motion, or to determine drug toxicity, drug mechanism. So here is how we went about, and I'll show you. First, I'll show you this movie. So this movie, it's illustrating what I'm talking about. So every dot you see on this plate here, on this image, is a culture that was printed for 15 minutes. So we just brought the cells together, 15 minutes, and we added retinoic acid. So high concentrated retinoic acid, you control on the bottom. This is the time of the experiment, and the bars is the size change. Watch what happens. Within 3 hours, you can see a dose dependence already. But look, the control without the drugs move much faster. And you have this dose dependence pattern. But which is very relevant, which is the new aspect of this measurement that we're doing here, is what comes next, which is the shape of these curves. The shape of these curves are very specific to the compound and to the cell type. So here, I'll go to the next slide. So how do we know it's killing? So the reason the high concentration is moving slower is because the cells are dying. And this is how we're showing that the cells are dying. So here's your picture at 5 hours. The control is smaller, high concentration is large, and this is just live dead staining. Green is live, red is dead. You can see it follows. And then we did MTT actually. We see the same dose dependence. And if we do the 2D comparison, the 3D is more resistant, which is expected. But what's really cool is, if you look at the compound, and this, we published this in scientific reports. So if you look at different compounds, here, pen of compounds, if you look as a collection, how these curves are different. This set of curve is different from this set of curve, from this set of curve. And again, the red in black here are flat because the cultures are not moving because the cells are dead. But can we use, the shape of these curves as a readout for biology? So here we have the same, before we had the same cell type with different drugs, which is treated 3 fibroblasts here. Now we have same drug, which is doxorubicin, chemotherapy, with 3 different cancer cell types: lung, liver. Look at the shape of this curve and say, okay, the blue is the high concentration. So we can actually differentiate the cell types, but they all show those sensitivities. So how can we effectively and visually differentiate the cell types besides just looking and saying, how can we quantify that? So what we did is basically we applied polynomial fit for these curves, okay? And from the polynomial fit, we can get the different cofactors of the fit. And then we use PCA analysis, principal component analysis, which is a multidimensional reduction technique it's part of the artificial intelligence machine learning portfolio. So now if you look at the parameters, the clustering of this data, liver, lung, and colon, we can clearly differentiate them and also in the dose dependence. So it's very powerful. Now, this data here, to get this data, we measured a full 384 plate every 15 seconds. That's really fast to measure the entire, and the only reason we're able to do that is because we use a mobile device. So this is the iPod, and so the iPod we have an app that takes a picture every 15 seconds, and we have the software that analyzes that. So most microscope cannot get even close to this imaging rate. So this is pretty cool. It's new, it's very powerful, and it works. So, and if you're not clear of what do I mean by this motion, this is a good example. So what we did here, don't worry so much about the red and green. So because it's the same cell type, this is a glioblastoma cells, and we magnetized them, and we brought them together, and we watch under microscope now. This is using a microscope, how the culture assembles. And this is the motion. That's that contracting that you saw on those wells. This is the microscopy. So watch what happens. See, they actually form a lumen, sort of a lumen, and it's a cup. So the cells are interacting, and this is 12 hours. So within 12 hours, it makes a spheroid. Which was cool here, we add T cell, which is fluorescent blue here, which is on the right, to actually see if we can see the infiltration at the same time that the culture assembles. And I'll show you the blue now channel. So you can actually see the T cells, CAR T, are the blue cells. I hope you can see on the video. So you can see it start flashing, the bright fluorescence. There's some background, but you can clearly see the bright fluorescence of the CAR T over time to start infiltrating the cells. Again, the CAR T are not magnetized. So again, it's an enabling tool. We'll look at this motion to see if we can determine CAR T cell targeting and killing. So it's a different way of looking biology. Look at this motion, and again, it's the first 12 hours. It's a very fast measurement, relatively speaking, instead of a week that you need for growth. And we have the dot. We made a spheroid, but we can also print rings. See, we can use a ring magnet and make a ring, like a wound healing type of assay. And we actually, in a collaboration with AstraZeneca, we use this for a vasoactivity assay, which is we show here. So why vasoactivity? Because in vivo vasoactivity measurement are very hard to do, and it's ex vivo. So basically, they take the aorta of the rat, we chop it up, and we stretch. And if the compound is vasoactive, if it's a vasoconstrictor that makes your blood pressure to go up, it's harder to stretch. If it's a vasodilator, a drug that makes you faint, it's easy to stretch. But it's not reproducible, it's not high throughput, and use animals. So our hypothesis was, can we take vascular smooth muscle cells, print a ring, add a compound, and look at this process? So here's the movie to show you. I hope you can see on the movie. So my mouse is not showing. Let me see if I can click on it. Oh, it's not going. Oh, there we go. So here, this image is straight from the movie. So this vascular smooth muscle, we use blebstatin. Blebstatin blocks phosphorylation myosin light chain. It's a vasodilator. So when we make the ring, the culture is going to tend to close fast. The vasodilator is going to slow down this process. So watch. So this is your ring. This is the image straight from the iPod. So this is the microscopy image of the ring. Watch what happens. So you have control on the bottom, high concentration of blebstatin on the top, and then your bar is here, and this is overnight. Look how it close. So, and of course, we can quantify that. We get the data, and we do the image analysis to get your curves for this mobility process. So it's pretty cool. And again, this we do all the imaging with the iPod. So that's your control and your dose dependence contractility. So that's the part of the cell mobility. And now I want to touch, because again, this is a bit of an improvised cartoon, but I want to plant a seed for using the magnetic 3D cell culture as a virus trap for different applications. So first, not so much the 3D cell culture here, but imagine a cell here. When you have a cell in 3D and you have a virus, you probably want access to them 360%, especially if you want to produce the virus rapidly. And when you have a monolayer, you have only half of the cell exposed. That's one thing. So now imagine if you can maximize this contact between virus and cells for vaccine production or testing a compound. You take your monolayer, you take your cells, you add the virus, and you mix the virus and the cells. The cells are magnetized, the virus, of course, it's not magnetized. Then when you place the cells in the magnetic field, the cells are going to trap the virus inside, even if it's for levitation or bioprinting. The levitation, if you're looking at air-liquid interface for long, that could be relevant. And then you magnetize the cells. Now, the beauty is you can transfer to a bioreactor, grow your virus. Now, a lot of times, you don't want to centrifuge your cells to remove the virus, the supernatant. Then you can use magnetic separation. You put on a magnet, you can remove your supernatant, and you can continue to grow, maximize use. So I just want to plant a seed of how you can use. Yes, we have a collaborator at Emory University that's starting to use this. And you have a few other groups in China too that are starting to use. So I just want to plant a seed to you guys, and we can do bioprinting. People say, oh, why is this bioprinting? We can use magnet different shapes and pattern them. This is a work we did. Actually, we implanted the magnetized cells back in the mouse. We differentiated. We did all the process using our system and actually magnetized we created a mini salivary gland, put it back on the mouse, and actually the culture, the tissue re-innervated and showed function. And the last slide I want to show is we did an experiment at the space station. This is Peg Whitson after she came back from her trip at National Geographic, and this is her in the ISS working with our system. These are magnets. These are the cultures. And if you don't know, cells in space grow in 3D because there's no gravity. So why do you need a 3D cell culture system in space? It's very hard to manipulate cells in space because you don't have gravity. The cells are floating around. So we send magnetized cells, and we were able to use the magnetic field to facilitate the process, to hold the cells, exchange media. And also, we tried to do the monolayer culture in space. Nobody had done a monolayer culture in space. So we wanted to print the cells on the membrane and see if we could create a 3D control in space, which was very interesting. And we're hoping to continue these experiments on a future trip to space station. the cells are stuck to a membrane, but they never spread and grow a monolayer. So we have a few ideas how we can try to use forces or magnetic fields as a surrogate for gravity to see if we can make them attach. So this is pretty cool. And of course, we have a lot of collaborators. We had funding from NIH, 3 different grants, CASES, Center for Advanced Science and Space, and we have a few big grants from NSF too, and a wide range of collaborators throughout the years to help this work. And hopefully I didn't go over time, but thank you very much, and I'll be glad to answer any questions. If you want a copy of the presentation, I'll be glad to send you the PDF of the presentation as well. Thank you very much. I'll answer questions live next.